we will discuss the uses of radioactivity and this radioactive substance called radioisotopes, and these radioisotopes are made in a nuclear reactor. In a nuclear reactor, these radioisotopes after irradiations produce, and these radioisotopes could be used in several places. And here in this session we will discuss that the radiation uses of radiation in sterilized equipment and smoke alarm. So first, the smoke alarm. A smoke alarm consists of a battery. This is the alarm system. A battery, an alarm device, and two ionization chambers which are identical, except for one being open to the air and other closed. So there are two ionization chambers, one is open to the air and the other one is closed. Each chamber contains two electrodes and a PD applied across them from the battery, and a potential difference is applied across the electrode. A small radioactive source in each chamber emits alpha particles from the both sides, both chambers. Alpha emitters are there, or that alpha emitter is actually the Americium-241. This is a radioactive source in a very small quantity, which produce ionization of the air molecules. So this ionize the air molecules, and that ion move toward the electrode and a current flow. If this radio alpha particles ionize the air in this vicinity, that produce ions, and that ion move toward the electrode and current flows. Since the conditions are the same in each chamber, the small current produced in each is the same. So the currents produced in each chamber will be the same. When the smoke, this is a normal condition, but whenever a smoke enters the chamber open to the air, the motion of ions is impeded. The motion of ion will be slow, because this smoke will have an interruption to this ionization process, or will obstruct the alpha radiation, and when they adhere to smoke particles, the current falls. So as a result of this, the current will fall, and the difference from the unchanged current in the closed chamber is detected electronically, and the alarm is activated. So this creates a difference from that closed chamber. This open chamber, a fall in current will appear, and it will create a difference from the closed chamber where the regular working was there. So as a result of this difference, it is electronically detected and alarm is activated. So alpha particles are chosen because they do not travel far in air. So why this alpha particle is chosen, why this Americium-241 is chosen? Because it do not travel. It doesn't travel, or they do not travel far in air, so do not pose a health risk. So its penetration is very low in air. A few centimeters, hardly a few centimeters can move. Even it cannot cross our dead skin. So a long half-life source is preferred for this smoke detection, radioactive source, so that the constant activity is maintained. If a low half-life source will be used, then this activity will not be maintained. After some half-life, this activity will decrease to the half value. So that's why a constant activity source is used. So the long half-life source is preferred. In the sterilization process, the gamma radiation is used. For a smoke detector, alpha radiation is used. For sterilization process, gamma radiation is used to sterilize medical instruments by killing bacteria. This alpha gamma radiation basically kills the bacteria. It is also used to irradiate certain foods. So this gamma radiation, it applies on the food, again killing bacteria to preserve the food for longer duration. This radiation is safe to use as a radioactive material goes into the food. The gamma radiation is chosen for its high penetrating power. So it will pass and it will move away. and a long half-life source is preferred so that a constant activity is maintained. So for this sterilization process, a gamma radiation is used. In the question number 9 from this Teach Yourself on page 273, explain how radioisotopes can be used to monitor the thickness of a metal sheet in industry. So radioisotopes can be used to monitor the thickness of a metal sheet in the industry. The thickness is a thickness gauge. If the radioisotopes are placed on one side of the moving sheet of a material and a GM tube on the other, the count rate decreases if thickness increases. If thickness increases, then the count rate will decrease. This technique is used to control automatically the thickness of the paper, plastic and metal sheet during manufacturing. So the thickness can be maintained by the count rate. If the count rate increases, it means the thickness is reducing. If count rate decreasing means if thickness is increasing means count rate will decrease. If count rate increases means thickness is decreasing. So this is very, very important. So we have to maintain a constant thickness. So we have to maintain this count rate, constant count rate, or a regular count rate. So this is basically used in paper, plastic and metal sheet manufacturing process. This is a system of radioactive gauging. Because of their range, beta emitters are suitable source for monitoring the thickness of the thin sheet, but gamma emitters would be needed for thicker sheets. For thicker sheets, beta is not suitable because that will be blocked by the thick sheets. So that's why gamma is used for the thicker material. So because they have a more penetrating power and are absorbed less. So the half-life of the source should be such a high or long so that its activity could be maintained for a longer duration, or a constant count rate will be monitored. So diagnosis and treatment of cancer. So to detect cancer, radioactive chemical that is preferably absorbed on cancer cells and emits gamma radiation. Gallium-67 is injected to the patient, and a gamma radiation is chosen because it easily penetrates skin and internal organs and has low ionizing power. So this gamma radiation can be used to detect cancer. So external imaging of the intensity of the gamma radiation indicates where the chemical has been strongly absorbed, and hence the location of the cancer cell can be identified. A radioisotopes should be chosen with a short half-life because the material is injected inside the body. So if its half-life is short, it will eventually decay, and the dose will be reduced exponentially. So for injecting or ingestion inside the body, a short-lived isotopes should be used so that it will not pose an ongoing radiation threats. The time needed to transfer the source from the production site to the patient also need to be considered. In the treatment of cancer by radiotherapy, high energy beam of gamma radiations are focused directly onto a tumor in order to kill the cancerous cell. In a radiotherapy, a high energy beam of gamma radiations are applied on the tumors to kill the cancerous cell. The beams are rotated around the body to minimize damage to the surrounding tissue. The beams are also rotated around the body to minimize the damage to the surrounding tissue. This is very delicate job. For external use, when a constant dose is required, a long half-life radioisotopes will be used. Tracers. The progress of a small amount of a weak radioisotopes injected into a system can be traced by a GM tube or other detector. This method is used in medicine to detect brain tumors and internal bleedings, and also used in the industry to measure the fluid flow in the pipe. So these tracers will be traced within the pipe, that the flow is a regular flow is available inside the pipe. Similarly, the carbon-14 dating, radioisotopes of carbon present in the air. This carbon-14 is taken in by the living plants and trees along with the non-radioactive carbon-12. When a tree dies, no fresh carbon is taken in. So as the carbon-14 continues to decay, and with its half-life is 5700 years, and the amount of carbon-14 compared with the amount of the carbon-12 becomes smaller. So with this years, its fraction will be reduced, and after 5700 years, its fraction will be half, and after 2 half-life, that is 11,400 years, its fraction will be 1/4 or quarter. So this is carbon dating to estimate the aging of the substance is used for archaeology. This is very, very popular to estimate